Welcome back to another video. In this module, we're going to discuss something called the complement system. This is another part of our innate immune response, which involves a broad and a little bit complicated system of antimicrobial proteins, all of which sort of work together or orchestrate a variety of immunological processes that control infection, especially against bacteria. There's a lot to say about complement, and I'm going to break this lecture up into two videos, but we'll start in this video by just sort of learning about the individual components of the complement system and how they begin to come together. So the complement system is not a single molecule, but many, and the molecules that make up complement are made in various locations in the body, but especially in the liver. So the liver makes a lot of the individual proteins that we associate with the complement system. It is secreted in the liver and then sort of enters the circulation through the veins coming out of the liver. And as you can see, I've just sort of drawn it here as these sort of green spheres floating around in the blood along with the red blood cells. And so a big idea here is that complement comes from the liver, but we find the molecules themselves highly enriched in blood. It's a major component of the blood. And when complement, these molecules that we call complement, when they encounter pathogens, a major thing that they do is stick to those pathogens. They sort of coat them, what you can see in this image of this bacterium being coated by these green molecules. And complement reactions can happen to bloodborne pathogens, but when we have infections in the tissues of our body, complement molecules can actually leave the circulation. They can sort of get out between the blood vessel walls, enter the tissues where they can sort of coat the pathogens in the tissues as well, and can also contribute to various antimicrobial properties. And we'll discuss the ways that complement actually sort of kills these microbes. But for now, we're really going to talk about how do they attach, how do they know what to attach to, and so on. So there are three major stages of complement activation in terms of when pathogens enter the body and complement first sort of detects them. And then in fact is the first stage, which is pattern recognition. Complement uses various strategies to detect non-self, just as many components of the innate immune response do. And we'll talk about some examples of how the complement system uses various molecular patterns to know to become activated and to initiate the response that will result in various effector mechanisms of pathogen control. Following detection of an infection, following pattern recognition, there is a pretty stereotyped enzymatic cascade. Many of the molecules that we associate with complement are enzymes, and more specifically, they are proteases. So we're going to see there's sort of a chain reaction of protease reactions, proteins being cleaved and then moving on to cleave another protein downstream of them. And this is just sort of how the system works, and we'll see that in more detail. Finally, as these enzymatic cascades proceed, the end result is going to be a variety of effector responses, meaning that they're going to influence the cells around them. These complement molecules are going to influence the cells around them to exert antimicrobial properties, things like phagocytosis, which you can see here. But the complement system can also kill pathogens directly through their own means, and we'll unpack what some of those strategies are in more detail as we go. But for now, I just sort of want to put the idea that there are three stages to this reaction, and we're going to start at the very top with pattern recognition. So to recognize infection, the complement system sort of has multiple tools at its disposal, multiple ways that it can go about becoming initiated. And we then sort of segregate complement reactions into three pathways. And these pathways are really defined by the way that they get started, the way that they initiate that initial stage of pattern recognition. The so-called classical pathway is initiated when complement binds to antibody, which itself is bound to a pathogen. So recall that antibodies bind to antigens, which are non-self entities on the surface of pathogens. And so in this scenario, the antibody is really doing the job of the detection of non-self. But molecules of the complement pathway, when they see that an antibody is bound to something, they use that as a kind of surrogate signal to say, if an antibody is stuck to it, it must be non-self, therefore we should attack it. And so the initiation of a complement response through the classical pathway involves interaction with antibody bound to pathogen. In contrast, another way that we can initiate a complement response is through something called the lectin pathway. Lectins are molecules that bind to sugars, and if you've taken a microbiology class before, you may know or remember that many pathogens have a lot of sort of unique and strange sugar residues on their surfaces. Microbes interact with the world through highly glycosylated proteins on their surface, oftentimes. Glycosylated meaning proteins that have been post-translationally modified with sugar molecules somehow. We can exploit the fact that pathogens have all of these sugars on their surface and that these sugars differ from the sugars that we typically find on our own host cells to recognize those microbial sugars as non-self signals. And the way that we do that are through specialized complement proteins, which themselves are lectins, sugar-binding molecules. So we initiate complement responses in response to microbial sugars through the lectin pathway. Finally, there's something called the alternative pathway, which is a little bit complicated when we don't yet have any context. But for now, I just want to say that rather than complement binding to antibody or to a lectin, in the alternative pathway, complement has means of attaching directly to the surface of a microbe itself. There'll be more to unpack about that, but for now, it's enough to say that the alternative pathway does not involve a kind of intermediate detection step. Complement just attaches directly to the surface of the pathogen, where it can then go about doing all the things that complement does, and we'll come back to that. But for now, let's start with the classical pathway, just because it's probably the most straightforward. I think that the classical pathway is a little bit of a misnomer because even though we call it classical, kind of indicating that maybe it's the oldest, or that it's the most fundamental somehow, the classical pathway is actually the newest pathway among the three, and that's because it uses antibody. And antibody is actually itself fairly evolutionarily recent. As I've said before, we only find antibody in vertebrates. The lectin and the alternative pathways are actually much more ancient, but the classical pathway is so named because it was the first to be discovered and described, and that's a very common feature of the nomenclature in biology. But because it was the first to be discovered and described, it's also it's sort of the most straightforward to learn about, just to kind of understand how complement reactions proceed. So let's jump into the classical complement pathway. So the complement system involves, no matter which pathway we're talking about, it involves a series of molecules which have been designated with the letter C and then a number. And you can see here on the slide that we have a molecule called C1, which is this sort of strange upside down umbrella shape, if you want to think of it that way. It's a combination of this kind of purple subunit with the orange and green subunits. This itself is collectively referred to as the C1 complex, or just C1. You can also see on the slide that there are molecules called C4 and C2. And in general, complement reactions proceed numerically from C1 going down the line. The only exception to that is that C4 sort of comes out of sequence. So we get C1, C4, and then C2. But from here on out, they tend to be in order. Let's start though at the beginning and talk about what C1 does. How does C1, is the first complement protein in the classical pathway, how does it initiate a response? Well, C1 binds to antibody, which itself is bound to the surface of a pathogen, is recognizing some antigen on the surface of a pathogen. And the component of the C1 complex that specifically binds to antibody is known as C1q, which is shown here in purple. So notice that it's the purple portions of this molecule that are interacting with the antibody. In addition to C1q, the antibody binding domain, there are additional subunits that we refer to as C1r and C1s. And these are the proteolytic domains. The active sites of C1r and C1s have protease activity. And the substrate of C1r and C1s are the molecules C4 and C2. And as a protease, the things that proteases typically do are to cleave things, and that is in fact exactly what happens. When C4 binds to C1, it is proteolytically cleaved into two cleavage products known as C4a and C4b. And this is another common thing, a theme that we'll see in the complement system, is that when something is cleaved in half, or I should say in two pieces, it's not directly in half, there's usually a smaller piece and a larger piece. One of those pieces is designated with the subscript A, and the other with the subscript B. And so here we get C4 being cleaved into C4A and C4B. Similarly, the molecule C2 is cleaved into two molecules, C2A and C2B. This video will be a little bit of alphabet soup. I'm going to do my best to sort of keep all terminology correct. But this is just one of those rites of passage in immunology. This is just a little bit of a kind of weird nomenclature for the complement system, but do your best to hang in there. So just to recap again, C1 cleaves C4 into C4A and C4B. C1 also cleaves C2 into C2A and C2B. For here, let's pause though. We've gotten this far in this complement reaction, and let's compare this initiation step in the classical pathway with what is a pretty similar set of reactions in the lectin pathway. So lectins, as I said, are molecules that bind to microbial sugars, and those are represented by these kind of branching green molecules on the surface of this imaginary pathogen membrane. And lectins, the two lectins that we could sort of most famously associate with the lectin complement pathway are molecules known as MBL, or mannose binding lectin, as well as ficalin. We won't go into detail about exactly what these two molecules bind, although you can tell from the name, mannose binding lectin, that there's mannose involved, which is a type of sugar. But essentially, you can see that they have a very similar structure to C1. They sort of have this upside-down umbrella shape. A key difference, though, is that MBL and ficalin as lectins, they also have proteolytic domains, but we refer to these as MASPs, M-A-S-P-S. And so the MASP domains of these lectin molecules are directly analogous to C1R and C1S that was found on the molecule C1. And they have exactly the same substrates, C4 and C2. So when C4 interacts with MBL or ficalin, it is cleaved by those MASP domains, again, into C4A and C4B. Similarly, C2 will be cleaved into C2A and C2B. And if you compare the last slide to this one, you'll see that this kind of right side of the slide, the proteolysis of C4 and C2 is exactly the same. The only difference is that initial molecule. The classical pathway uses C1, whereas the lectin pathway uses lectins like MBL and ficalin. But everything downstream from here looks exactly the same. And so if everything is exactly the same, let's go one more step downstream and talk about where these two reactions end up, or at least where they end up next. So on this slide, you can see on the top is the progression of a classical complement reaction. On the bottom is the same reaction in the lectin pathway. Notice that in the classical pathway, we're starting with C1 bound to antibody. In the lectin pathway, we're using a lectin like MBL to bind to a microbial sugar on the surface of the molecule. And we can see again that C4 and C2 are being cleaved into those cleavage products. Notice now, however, that C4A and C2B, kind of the smaller of the two cleavage products, you can kind of see that they're floating away. We're getting rid of them, which is what this flow chart is indicating. And for the most part, we are not really going to use them any further, at least for now. But the larger of the two cleavage products, C4B and C2A, they stick around and they actually come together to form a new molecular complex that we refer to as C4B2A, all together kind of as a single word, C4B2A. This is essentially a new molecule. It forms a new quaternary structure, a macromolecular complex, which itself now takes on new enzymatic activity. C4B2A, the complex, is an enzyme. And that enzyme, its activity is something that we call C3 convertase activity. As a C3 convertase, the substrate of the C4B2A complex is the molecule C3. And as you might guess, based on what we've said so far, the C3 convertase is also a protease, and the reaction that it catalyzes is the proteolysis of C3 into C3b and C3a. And again, you can kind of see that C3a is smaller and kind of floating away. C3b is larger. And as we'll see in the next lecture, we keep this C3b and use it for additional purposes downstream. It's very important to note that fundamentally, after this initial step of pattern recognition by either C1 or MBL, both pathways make a C3 convertase, and they make the same C3 convertase. In both examples, the C3 convertase is C4b2A. And so we can say that all complement pathways converge on the formation of a C3 convertase. It won't always be this exact C3 convertase, but the classical and lectin pathways share the same one. And so basically everything that's happening downstream is going to look exactly the same for the classical and the lectin pathways. Okay, so we've made C3b and C3a. Some stuff is going to happen downstream, but now let's back up and talk about how the alternative pathway works. How do we get this same set of reactions occurring in the alternative pathway? It looks quite a bit different. So the alternative pathway can be initiated through two mechanisms or two means. One is that the alternative pathway can exploit existing C3b that has been generated by either the classical pathway or the lectin pathway. So they're already going on kind of in the background. They're forming a C3 convertase, creating C3b, and some of that C3b can then be used to initiate the alternative pathway. And this functions as a feed-forward mechanism, whereby we start a complement reaction and we have a little bit of complement C3b being made, and then we can sort of kick in this sort of side arm of the pathway, the alternative pathway, to then create even more C3b. So the way that happens is that C3b attaches to the surface of the microbe when it's made, and it itself then binds to a new factor that we call factor B. So that's just designated with the letter B here in this diagram. Factor B, when it is bound to C3b, forms a new complex called C3b, and that serves as a substrate for an additional enzyme in the complement system, particularly in the alternative pathway, called factor D. Factor D is a protease. Its substrate is the molecule B, and it can only bind to factor B when B itself is bound to C3b. And that's kind of what we're trying to indicate with the complementarity of these shapes. Notice that these shapes are drawn in a way that D is binding to the combined shape of both C3b and factor B joined together. However, when these molecules successfully come together, the protease activity of D is engaged, and then factor B is cleaved into two molecules, BA and BB. So just like when we cleave C3, we get C3A and C3B. When we cleave factor B, we get two cleavage products, BA and BB. And notice the capital B, lowercase a, capital B, lowercase b. BA floats away again, and we keep that larger subunit, which is designated as B. And this forms a new molecule that collectively, again, we refer to as C3bBB, as all one word, all those letters just mushed together. C3bBB is now its own protease, and it, in fact, can serve as a C3 convertase. And so this molecule serves as one of the possible C3 convertases of the alternative pathway. And so as a C3 convertase, C3bBB can bind to new molecules of C3, new uncleaved molecules of C3, and cleave them, forming new copies of C3b and C3a. And again, we can then use these cleavage products perhaps to make even more complement by restarting this process. Or we'll see that there are other effector mechanisms that these molecules can mediate, but we'll come to that point a little bit later. So I should say that on the top here, this is the alternative pathway, and this is the way that we start the alternative pathway, again, by using existing copies of C3b that have already been produced by other complement reactions. So this is more about feed-forward amplification. However, we can have spontaneous initiation of the alternative pathway, meaning that it can happen de novo without complement reactions having already happened. And the way that we get a spontaneous initiation of the alternative pathway is through something we call the hydrolysis of C3. You may remember that hydrolysis is when we sort of cleave or we attach a water molecule to something. And so when C3 is hydrolyzed, when water is inserted into it, that forms a new molecule that we designate C3H2O, kind of all as one word again. And C3H2O itself can also bind to factor B. And just like here, where we see that the combination of C3b with factor B allowed factor D to bind, similarly, factor D can bind to the combination of C3H2O and factor B. When that occurs, D is able to cleave factor B into BA and BB, and this is how we can get the second type of C3 convertase that we find in the alternative pathway. That is known as C3H2OBB. Functionally, C3H2OBB is identical to C3BBB. They are both C3 convertases. The only difference is just sort of how we initiate them, what this side of the molecule looks like. But in either case, they can both bind to new uncleaved copies of C3 and cleave it into C3A and C3B. It will then go on to do the next steps in the complement cascade. So again, here we're talking about the alternative pathway in two different ways that it can proceed. But I want to re-stress the point that no matter which pathway we're talking about, our main goal, or at least the main first goal, is to create a C3 convertase, because the cleavage of C3 is what really unlocks everything that comes next in the complement system, and that will be the focus of our next video. But for now, let's just summarize this part one of our journey through the complement system. So we learned that complement proteins are a really ancient and evolutionarily conserved antimicrobial defense system, and they are able to recognize various non-self entities such as bacterial sugars, or indirectly they can recognize non-self through interacting with antibody, which itself has coded a pathogen. And one thing that I did not go into is that host cells are protected. There are various mechanisms that prevent complement from binding to host cells and target it only to bacteria and other types of pathogens that we can recognize as non-self. There are three major pathways for complement reactions. Another term that I haven't used yet is another commonly used term, fixation. We say that complement fixes itself to the surface of pathogens, or sort of meaning attaches to the surface of pathogens. And these three pathways of complement fixation were the lectin pathway, which recognizes microbial sugars or carbohydrates, the classical pathway, again, which recognizes antibody, as well as the alternative pathway, which can either begin spontaneously or through amplification of C3b being produced by the other two pathways. Finally, we highlighted the point that all three of these pathways converge on the generation of a C3 convertase. And some general themes of the biological reactions that were happening is that many complement proteins require proteolytic cleavage to be activated, and that those cleavage products themselves have intrinsic protease activity. And there's really a chain reaction of things cleaving each other in sequence. And we'll see that theme continue to play out in the next video, where we really focus on the effector mechanisms of the complement system. See you there.